I'd like to go ahead and start and introduce Janet Jansson from Pacific Northwest National Laboratory, and she's going to be talking about soil microbes on Earth and in space.
So my title has changed. Since I've been at this meeting, I've been learning a lot, and so I'm trying to put more of a perspective into the space angle and not talking entirely about climate change on Earth. So my talk will be on soil microbiomes on Earth and in space, implications for space. The picture here is me standing in our local field site in Washington State, where we've set up an irrigation trial to study drought in that system. Next slide. My little thing doesn't work. So this is something I'd like to use to introduce the complexity of the soil environment. And so this is a quote from the 1500s from Leonardo da Vinci. And even back then, he said, we know more about the celestial bodies than the soil underfoot. And so this is a telescopic view of stars in our galaxy. The numbers are huge. If I could have the next slide. If we then go into a microscopic view of soil, the numbers are also huge. We're just looking at a different scale. And so this is what I call the microbial universe, or Earth's dark matter. So here, a single teaspoon of soil has billions of cells. Most of them have never been cultivated, and it's difficult then to understand their functions. Their properties aren't known. And it's really important because these microorganisms are vital for functioning on our planet. Next slide. So I had to put this one in because, okay, soil's important. Matt Damon was able to make soil, and this is a good combination, a link between the human microbiome and the soil microbiome. If anybody has read the book or seen the movie, you'll know what I'm talking about there. But soil is vital for growth of plants and for degradation of organic carbon compounds and pollutants. You could have pollutants on Mars or on the space station. They're also largely responsible for cycling of nitrogen and other essential nutrients. They're a source of biodiversity, and they're also, well, for example, source of new antibiotics and other drugs. And so we're just beginning to explore, to scratch the surface of this really unknown microbial world in soil. Next slide. So I tried to think during the meeting of some opportunities for space research when considering the soil environment. So on Earth, we know that soil microorganisms, they reside in communities. They function as interacting members of consortia. And they've evolved over billions of years to be able to utilize the nutrients that they have available and also to cope with stress. And so what happens in space if we take these organisms, these communities of organisms, and they're exposed to a space environment? Do they still interact in the same way? Are they still able to cycle nutrients and function in the same way? Another thing is that modeling combined with experiments in defined consortia where you know the members, you know the membership, is really a useful tool. And this is something that I'll be talking about a little later, to determine how the soil microbiome will react to unique conditions. So you can perturb the system and look at how the communities interact. And so what happens in space with, for example, with microgravity? And then finally, this knowledge, once we understand how these microbial communities interact in a certain context, can we harness those potential beneficial properties for targeted endpoints in space? For example, biodecomposition, nutrient cycling, and plant growth promotion. Next slide. So this is another thing I was thinking about during this meeting. I didn't realize, or it hadn't really hit me, that there probably isn't matrix pressure. The hydrostatic pressure is not there in space. For soil communities, this is really a vital process because soil microorganisms, they inhabit these pores that are very discrete in soil. I don't have a pointer. So, thanks. Yeah, I think this will work better. So if you think of, if you blow out this this microbial view of this pore space, the water content is going to govern how the communities interact. And I'll be showing several cartoons that use this color scheme with pink for bacteria, these little yellow viruses, and green fungal hyphae. And so here you can see, for example, this, if you have a discrete consortium here and one here, the water is going to govern whether they're able to interact or not. And so you can have diffusible, in a case where you have more water, you can have metabolites and cells even diffusing and moving back and forth. But as it dries up, these become isolated and more discrete. Here I have a fungal hyphae that's connecting too. So if you don't have hydrostatic pressure or matrix potential in space, how is that going to influence these discrete consortia that you have in a soil habitat? I have absolutely no idea. I would love to discuss it if somebody knows. Oh, next slide, please. So I'm going to give, originally I was going to talk about climate change primarily, but I decided not to. I'll give a few examples of climate change impacts in soil microbiomes, permafrost, thaw, and drought. I'm going to also give some updates on some of our omics platforms. And really here the aim is to be able to study microbial interactions and biochemical reactions in soil. I'll also just highlight one of our new virome pipelines that we developed. And then I will talk about evolution of naturally interacting reduced complexity soil consortia. That's a resource for understanding microbial interactions and could be a very valuable resource for space as well. Next. So we are experiencing climate change. The Arctic is particularly impacted by warming. And so as the warming trend continues, the permafrost is starting to thaw. We're seeing coastal erosion, landscape degradation, these collapsed bogs forming thermokarsts. And what is going to happen to all of that carbon that is stored in permafrost? Permafrost is a huge reservoir of soil carbon. Yeah, and as it's being degraded, that can produce more greenhouse gases, CO2, and methane. Next slide, please. So this is a close-up view of a permafrost environment. So we have the permafrost that's defined as permanently frozen soil. Has to be frozen at least 2 years to be classified as permafrost. And so here we have a lot of frozen organic matter. Then the active layer on top of that is seasonally frozen and thawed. And that's where a lot of the microbial respiration and metabolism occurs. And then here you can see, depending on the microbial processes, you have CO2 or methane being produced. Next slide, please. So just a few highlights. We've studied this for many years. Most of this is published, but a couple of highlights. So one of our first big studies of permafrost was to do an incubation in the laboratory of frozen permafrost and then over a short term subject it to thaw and see what happened. And this was using a metagenomics approach. And I like this graph. It was one of the earlier examples of using soil metagenomics in this way. And so what it's showing is in red, these are pathways that are dominating under the thaw environment. And in blue, those are pathways that are more represented in the frozen environment. And so after a very short thaw, a period of a week, this flips over. And so the pathways, the metabolism, the organisms that were represented in permafrost become a lot more similar to those that we see in the active layer. And so it's a very rapid response to thaw. This was also at the beginning of being able to get genomes out of metagenomes, and we were able to describe a novel methanogen genome from the metagenomes, which was super cool. Next. Okay, so this, by the way, is a picture I took of an iceberg on the coast of Greenland, and that was also amazing, by the way. But it's really the tip of the iceberg is to identify microorganisms. We really need to understand more than that, the function. And so, next, please. So the key question is, what are they doing? We want to assess the activity of functional genes and the proteins that are produced. Next. So this is another way of thinking about that. Currently, we can sequence anything, so we can get information about the community composition and the gene composition in any habitat. So I used to think of soil as a black box. I'd say now it's more brown because we know who's there. So next. But we still don't understand that much really about the function. So the genes, proteins, and metabolites, for example, that are involved in carbon and nutrient cycling, those are very gross processes. We can measure gas coming in and going out and know that respiration is occurring, but all of those processes really are not well characterized. And so next, where we want to go is into more of a brown box for function, where we can understand which pathways are present, what's being expressed, and being able to eventually control these metabolic pathways for certain endpoints. Next. So another concept is that we need to go beyond genotype to phenotype. So in this little cartoon, this cell has a genotype, and that and of course a phenotype. And so examples of cellular phenotypes include everything from motility, antibiotic resistance, et cetera. And the phenotype is also governed by which genes are expressed into RNA, proteins, and metabolites, and everybody knows this. And so we can understand the genotype to phenotype. Next. But for a community, we really need to understand the metaphenotype. And so now we can get metagenomes. So metagenomes are the community genomes. So that's the metagenome. And the phenotype is governed by the environment, the factors in the environment that govern what is expressed. And so that results in a community phenome or metaphenome. And this is really where we want to go to understand the functioning of the system. Next. And again, this is just illustrating that for a metaphenome, we want to go from gene content to regulation, expression, protein content, and metabolomics to be able to really understand the metaphenome of a community. Okay? Next. Oops, back one. So this is another paper where we were looking at permafrost, but using more of a multi-omics approach to determine function and what happens during thaw. In this study, we looked at a natural thaw gradient starting from a frozen permafrost to a naturally thawed active layer to a collapsed bog. And so here, this graph is comparing the metatranscriptome to the metagenome. This is looking at functional genes that we have in the metatranscriptome compared to metagenome. And that's a nice way to assess who's more or less active in a different sample type. So the permafrost is blue, active layer green, and the bog is in pink. So on the bottom, I know this is maybe hard to see, but these are the proteome data. And so we had matched metagenomes and proteomes for each of these types of locations, from permafrost to active layer to bog. And then we're able to key in the proteins that were expressed under these different conditions. So in the permafrost, this is the first time anybody had really tried to look at functional activity using this approach in permafrost. And we were really surprised to see that there were a lot of pathways for iron reduction. This is something that we had not hypothesized would be occurring in that system. And so we went back and looked, and sure enough, there were iron reduction processes in the frozen permafrost that was happening in that environment. As we transition into the active layer, we have a lot of proteins for central carbon metabolism because the carbon is starting to be catabolized, a lot of aerobic processes. And in this thermokarst bog, it's anaerobic, and we see that the whole pathway for methane production and that's this pathway here. And so the nice thing about this is we can see the transition in function as you go across this thaw gradient. Next. Also, another thing that we're doing now is using the metagenomes to bin genomes. I know a lot of people are doing that now. I think that's really fun because we can use the genomes to understand the operon structure and the pathways. Like in the good old days when we were looking at genome isolates. We can now do that in metagenomes without having to isolate the organisms. And so this is an example of a bin for methanogen. We can see the pathway in the, this is a different study, but again, going across a gradient. In this case, it's a hydrological gradient, but we can see different kinds of genomes and different kinds of pathways for metabolism and hone in on some of the things that we're interested in. Next. Okay, so I'm going to briefly talk about drought. So drought, we're focusing on different grassland ecosystems when we're looking at drought. Climate predictions predict that in the future, a lot of these areas in the United States are going to be experiencing more drought. And this is just showing the darker the brown color, the more drought that is anticipated to be happening. And this is looking at the upper surface layers. So we anticipate mainly more drought in the future. This is important because these areas where we have the grassland soils, like the prairie soils, they contain a lot of carbon also. And so as the soil dries down with drought, how does that influence the carbon cycle? Next. So back to this cartoon, which is what we originally developed it for, where we want to understand how changes in soil moisture are going to influence the connectivity between these discrete microbial consortia, and they're able to interact. We're also interested in interkingdom interactions between bacteria, fungi, and viruses, and archaea, and how they interact to share metabolites, for example. Okay, so this is one of our field trials that we set up. This is in Kansas, and we collected soils from 3 different locations across the field. So it's A, B, and C. And then it was a very simple experiment. We either saturated the soil and incubated, or we dried it and incubated for 15 days, and then we ran it through our omics analysis. Next. Okay, so to summarize what we found, in all 3 cases, we're looking at 16S data, so community composition. We also did the metagenome comparison, but it was the same. The transcriptome, expressed genes, and the metabolites in this case, and the different shapes of the different soil locations. So the soils, the different soil locations clustered together, irrespective of treatment, but we do see this differentiation between soil drying, so a drought scenario for all across these different levels of expression. We don't see as large of a difference with wetting, and that was something that we didn't expect. We thought that there would be a larger response to soil wetting. So you can see the wet soils are similar to the controls in most cases. And so we think that historically, these soils are more used to having a wetter scenario, and so they didn't shift as much as when we desiccated it. Next. So when we look at the metabolites in more detail, we can see that there are many that specifically shift or are up, in this case, in the dry soils. And if you click one more time, if we just look at one of these as sucrose, I would say this is significantly higher. I don't know if I dare to, but at least across all 3 soils, it looks a lot higher. and it also makes sense because this is an osmolite, and it protects cells as they dry and are desiccated. Next. So you can map this data. This is what we're currently doing onto networks. So this is a network that contains both the metabolite data and the transcript data, and it's looking at across the different soil locations. with the aim to see, are there signatures of that state change, signatures of a dry soil? And here we can see these larger red nodes are pathways or metabolites that are consistently higher in a dry state. And that is really nice because then we can start to make more hypotheses about what kinds of changes we would expect in bacterial metabolism under a drought scenario. And this is just focusing in on one of the pathways. This is for tree hallose production, which is higher in the dry soils, or degradation, which is lower in the dry soils. It's higher in the wet soils. And so we can nicely see the pathway in this complex community. So it's not just a single organism, it's the whole community response to drying. And tree hallose is another osmolyte, an osmoprotectant compound. Next. So these are our three soil grasslands that we're looking at right now. And we chose them because they experience different kinds of rainfall or irrigation inputs. So our site is super dry. It's a desert environment. It's either dry land farming or irrigation, which is great for setting up an irrigation field trial. Iowa is rain-fed and tile drained, so it's a wetter soil. And then Kansas is somewhere in the middle, which can be irrigated. And we have almost 2 terabase metagenomes for each of these locations. That is really a blessing, but it's mainly a curse, I can tell you, because it's so hard to assemble those metagenomes. We weren't able to do it using our supercomputer facility at PNNL, so we had to collaborate with some other folks at NERSC, and they had a new pipeline, and this was their test case for assembly of soil metagenomes, and it took a long time, but we now have it assembled. Next. So these are the kinds of things we want to explore in those grasslands. We want to understand the fundamental principles underlying these microbial interactions. So interactions between microorganisms, and that could include everything from biomass turnover to production of these polysaccharides or competition, and also the molecular interactions, and that could be syntrophy, quorum sensing, which we discussed yesterday, production of antimicrobials, and also division of labor. Next. So what we first did was map the metagenome. Did I do that? What we first did was map the metagenomes onto these biochemical reaction maps. And so this is Washington, Iowa, and Kansas. And you can see just without thinking about the detail that they look different. And so we're seeing different kinds of pathways, the potential for metabolism in the 3 different soils. And they're color-coded by the amount of reads that we have in these different metagenomes. So the redder colors means that there are more of those pathways present, and the bluer colors, they're there, but they're not as dominant. And then in these cases, we don't see much evidence of those pathways being there. Next. So we have the potential metabolism. We're interested in these interkingdom interactions. There's very little known about soil viruses, but when we have these large metagenomes, we can explore them for viral signatures. So we developed a new informatics pipeline tool. We call it VIR funnel. And so we have a different sorts of algorithms that are analyzing the data and validating the data. And at the end, we're ending up with highly confident predictions of viral sequences. And this is an example of one of the ways to display that information, which is not very useful, actually. But this is a clustering of the viral data. But if we go to the next slide, if you hone in on specific regions of these clusters, it starts to get really interesting. So here, these are coded by source soil. So purple, Washington, orange is Iowa, and blue is Kansas. We have some reference viral sequences, which are in gray. And then the different kinds of informatics tools are either circles or triangles. But you can see that just in this little snapshot from that huge hairball, we can find some clusters that are soil specific, as this one is only in Kansas. We can find some that are, these viruses are in both Iowa and Washington, and this cluster contains sequences from all 3 soils. And here's one, again, only found in Washington. And so this is the sort of thing that we're really interested in right now, developing further. Next. Also, when you have these viral sequences on contigs, most of them look to be complete, or many of them look to be complete. You can mine the contigs for auxiliary metabolic genes that might be of interest, that are not typical viral genes. And this is just one example of a chitinase gene, or chitosanase gene that we found in a lot of the viruses in soil. And these are not normally found in other kinds of viral sequences. It seems to be specific for soil viruses. And they cluster in a phylogenetic tree. So the viral sequences are green, or no, orange, excuse me, I can't see it. The viral sequences are orange. And the green are the bacterial sequences, and the purple are fungal chitosanase sequences. And these are just examples of scaffolds where we have that gene present in, and the rest of the genes are typical virus genes. So and this is just one example. We're finding a lot of interesting auxiliary metabolic genes in the viruses that are involved in carbon cycling, potentially. This one we did express, and we found out it's an active chitosanase. So next. So how much time do I have? I have a couple of minutes. A couple minutes? Okay, that's fine. So the last thing I was going to talk about is development of these reduced complexity model soil consortia. So we took a couple of approaches. We either developed them in irradiated sterile soil, so you have the soil matrix, or on plates where we have a chitin substrate. And then these consortia, we wanted to have 20 to 200 naturally interacting species, were then tested for their ability to be stored and then reconstituted. Next. So this is a lot of data, but this is showing for both the bacteria and for the fungi, the changes over time. So these are 60 nests here and ITS here over 15 weeks on the top, and I think it's 11 on the bottom. So we made a series of dilutions, either in liquid media or in sterile soil, liquid media or sterile soil. And in the end, we were interested in seeing if we could get a representation that was similar to our source soil. And those are these narrow bars at the front. Next. So we did find that we got a decrease in richness over time. And this is the liquid media results here, and these are the results in soil for the bacteria. And I'm going to have to go kind of quickly through this. So if we go to the next slide, this is also showing the similar thing for the fungi. Next. And what we really wanted to see is if we were getting a reduction that was stable over time. And so this is a way to look at changes in beta diversity between successive time points. And you can just see that there's a lot of jumping around in the beginning, but we start to stabilize at the end. So this is, you can see, as the measure of volatility or stability. Next. And this is for the fungi. Next. So we were able to get several of these model consortia. We tested their ability to be stored and reconstituted in glycerol or by lyophilization. So if you just look at this one here, the triangle is the original sample, the consortium, and these are the ones after reconstitution, and you can see that they're quite similar. That's not always the case, but in this particular consortium, this brown one, it seemed to work very well. So we're focusing on that consortium. We know the composition. We have some extended cultures, and this is some examples of the community participants that are in that consortium. Next. And finally, we can also, once we have these model consortia, we can study them in a different ways. If you're interested in spatial interactions, we have a soil chip that allows you to look at at the way that the species assemble, their interactions, etc. If you're interested, I'd be happy to talk about it later. So we can go on to the end. Next. So the big picture, we live on a microbial planet. This is from the Earth Microbiome project that Rob Knight, Jack Gilbert, and I published a couple years ago. And so we know our planet is undergoing change, and those have largely unknown consequences on microbial functions that are essential for life. So the next challenges for our planet and beyond are to discover how organisms interact, use that information to make better models, predictive models, and finally, we can use that to control the microbial communities for certain functions. Next, I'd just like to acknowledge my team, the SFA team, soil SFA team at PNNL, and thank you for your attention in the last slide.